Spirogera and Rhizopas, all important bits. Now we discuss about the plant kingdom examples. Bryophyte example Funaria, Pteridophyte example Pteris, and Gymnosperm example Cycas. Funaria is a bryophyte where the gametophyte is dominant, so sexual reproduction appears first, then asexual reproduction. Pteris is a sporophytic generation is dominant, so first asexual reproduction takes place, then sexual reproduction occurs. Cycas also sporophytic generation is dominant, first formation of spores, then formation of gametophytes takes place. Funaria shows two stages in its life cycle. One is a juvenile stage called protonema and an adult stage called gametophore. Gametophore shows root-like rhizide, stem-like colloid, leaf-like phyllite. On the lateral branches, foot, seta and capsular form. Capsule is diploid, but it is surrounded by a cap-like structure called calyptra, which is haploid. Why calyptra is haploid? Because it develops from ventral wall of archegonium. Then Funaria becomes monoecious, dioecious. Then antheridial cluster, male flower, archegonial cluster, female branch. Formation of gametes, fusion of gametes, as Funaria is a bryophyte, completely depends on water for fertilization. Then Pteris, some important features. Then Cycas, some important characters, and we discuss some important bits in Pteris, Funaria and Cycas. Some important bits regarding, in these bits we can come across all the important aspects regarding Funaria, Pteris and Cycas. Match the following in Funaria. Match the following in Funaria. List 1. Uniseriated with chloroplast. You know what is uniseriated? If a single layer is there, cells are arranged like this. This is called multicellular uniseriate. So these cells are arranged like this and they are in two rows. So this is multicellular. All are multicellular uniseriated. This is multicellular biseriated. Cells are arranged in two rows. This is multicellular multiseriate. Multicellular multiseriated. Seriated means the number of rows. If only one row, uniseriate. If two rows, biseriate. If three rows or more than three rows, it is multiseriate. So here uniseriate with chloroplast, uniseriated without chloroplast, single layered with chloroplast, single layered without chloroplast. Answer is 3. Uniseriate with chloroplast is 2. Paraphysis. Paraphyses are special hair-like structures intermixed with antheridia and archegonia. Antheridial paraphysis shows subapical apex, whereas archegonial paraphysis shows pointed tips. They contain chloroplast, perform photosynthesis, provide protection to antheridia, store water in between themselves as mucilage. Then uniseriated without chloroplast or neck canal cells. Second B is 5. Then single layered with chloroplast is epidermis of colloid. Epidermis of colloid is single layered with chloroplast. Single layered without chloroplast is antheridial jocket. Antheridial jocket is single layered but biseriated cells, but it is without chloroplast. So answer is A2, B5, C3, D1. Now this is a assertion reasoning question. Funaria is called myxohydric mass is correct. Funaria is called myxohydric mass is correct. Funaria conducts water internally through capillary channels and externally by hydroids. Funaria is a myxohydric mass is correct. Funaria conducts water internally through capillary channels and externally by hydroids is wrong. The answer must be Funaria conducts water internally by hydroids and externally by capillary channels. But here it is given in a reverse direction. Funaria conducts water internally through capillary channels and externally by hydroids. So Funaria is a myxohydric mass. Myxohydric mass means it conducts water internally by hydroids, externally by capillary channels. So A is correct, R is false. So Funaria conducts water internally through capillary channels, externally by hydroids is wrong. Next, arrange the following of funereal archegonium in ascending order. Ascending order means less number to more number. Number of layers in the jocket of venter, number of vertical rows in the neck jocket, number of cover cells, number of venter canal cells. In any archegonium of Pteris, in archegonium of Funaria, the archegonium will be like this. It shows a multicellular multiseriate stalk. Venter surrounded by two-layered jacket. Venter surrounded by two-layered jacket. This venter encloses one venter canal cell and one egg cell. It encloses one venter canal cell and one egg cell. Neck is very short, made up of 6 rows of neck cells. 1, 2, 3, 4, 5, 6 rows of neck cells. Then 6 or more neck canal cells with 4 opercular cells. This is the structure of archegonium of Funaria. So here the answer is 2. First comes number of venter canal cells. Number of venter canal cell is only 1. Number of venter canal cell. Then comes 1. Number of layers in the jacket layer of venter is 2-layered jacket. So 1 and 2. Then number of cover cells. Number of cover cells will be, cover cells or opercular cells will be 4. So 1, 2, 4. Then next is 2. Number of vertical rows of neck jacket. Number of vertical rows of neck jacket will be 6 rows of cells. So 6. So 1, 2, 4, 6. This is the answer because it is ascending order. So the question is, I will repeat this question. Arrange the following of Funaria archegonium in ascending order. Number of layers in the jacket of venter is 2. Number of vertical rows in the neck jacket is 6. Number of cover cells is 4 and number of venter canal cells is 1. So answer is 4, 1, 3, 2. So second is the option. This is an assertion reasoning question. Funaria gametophore is monoecious and autoecious. Funaria gametophore is monoecious and autoecious means, monoecious means both male and female reproductive structures present in the same plant. Autoecious means both male and female reproductive structures present on different portions of the same plant. That means male reproductive structures antheridia present at the tip of the main axis. Female reproductive structure archegonia are present at the tip of lateral branches. So Funaria gametophore is monoecious and autoecious. Reason: male and female sex organs developed in groups in the same plant on different branches. Because in the same plant it is monoecious. Because on different branches of the same plant it is autoecious. So assertion and reason both are correct. Reason is the correct explanation of A. So answer is 1. Identify the correct sequence related to development of antherozoids in Funaria. Development of antherozoids in Funaria. This is an antheridium in Funaria. Antheridium shows a club-shaped structure with a multicellular biseriate stalk and a globose body. Body is surrounded by single layered jacket and it shows 2 special opercular cells or lid cells. Opercular cells or lid cells. Inside the body you will find special androgonial cells. These androgonial cells divide and produce androcyte mother cells. Androcyte mother cells. Androcyte mother cells divides and produce androcytes. Every androcyte mother cell produce 2 androcytes. These androcytes does not divide, they just metamorphose. They just metamorphose to form 2 sickle-shaped biflagellate motile antherozoids. Sickle-shaped biflagellate motile antherozoid. So identify the correct sequence related to development of antherozoids of Funaria is 4th option. Androgonial cell, androcyte mother cell, androcytes, antherozoids. It is not androcyte and androcyte mother cell, androgonial cell. It is not antherozoids, androgonial cell, androcyte mother cell. It is not androcyte, antherozoid, androcyte mother cell, androgonial cell. But the correct sequence is androgonial cell, androcyte mother cell, androcytes, and every androcyte metamorphosed to form 2 antherozoids. So sequence is androgonial cells, androcyte mother cells, androcytes, and antherozoids. So answer is 4th one. Assertion: the sporogonium of Funaria, diploid semi-parasitic, physically attaches to adult gametophore. The sporogonium is sporophyte. It is diploid, definitely diploid, and it is a partially parasite, semi-parasite, physically attached to the adult gametophore. It is attached to the lateral branch of the adult gametophore. Reason: zygote in venter undergo mitotic divisions, form embryo, later develops into sporogonium. So, zygote is present on the lateral branches. Zygote is present on the lateral branches. It develops into sporogonium, and it first develops into embryo, later develops as sporogonium. Because zygote is present in the venter undergoing mitotic division, forming embryo which later develops into sporogonium, the sporogonium of Funaria is not semi-parasite. Because it depends on the gametophyte only and for water and minerals, it takes food, it can synthesize its food, it is semi-parasite. So, both A and R are correct. R will not be the correct explanation of A. So, second option will be the answer for this. Identify the incorrect one regarding Calyptra of Funaria. It is haploid, correct. It develops from venter jacket after fertilization is correct. It belongs to the gametophytic generation is correct. Calyptra is always haploid. Why it is haploid? Because it develops from the venter jacket after fertilization is correct. And because it is haploid, it belongs to gametophytic generation is correct. It functions as a transpiring shield around mature sporogonium is wrong. It functions like a transpiring shield in young sporogonium is correct. Mature sporogonium, Calyptra is gone, operculum is gone, and peristomial teeth get exposed. So, it functions as transpiring shield around the mature sporogonium is wrong. So, first option is wrong here. Identify the ploidy of the following: Protonyma of Funaria, sporogynous cells of Funaria, pericytium of Funaria, apophysis of Funaria. It is protonyma of Funaria is N. Sporogynous cell of Funaria is 2N. Pericytium of Funaria is N. Apophysis is 2N. Because apophysis is present in capsule, it is 2N. So, protonyma of Funaria is haploid N. Sporogynous cell of Funaria is present in the sporophyte, hence it is 2N. Pericytium of Funaria is present surrounding the archegonial cluster. So, it is present in the gametophyte, so definitely haploid. And apophysis of Funaria is present in sporophytic capsule, so it is 2N. Answer is N, 2N, N, 2N. Third option. Choose the incorrect statement with reference to Funaria. Columella degenerate at maturity, correct. Columella degenerate at maturity is correct. We have to identify incorrect statement. Rhizidal branches can convert into chloronemal branches on exposure to sunlight. Definitely when a rhizidal branch is exposed, comes out of the soil, it gets sunlight and converts into chloronemal branches. Seta also helps in dispersal of spores. Definitely after peristomial teeth, the twisting and jerking movements of seta helps in the dispersal of spores. So, the incorrect statement here is endostome is attached to the rim and exostome is attached to the annulus is wrong. Both endostome and exostome, that means peristomial teeth, are attached laterally to the rim or diaphragm. So, they are attached laterally to the rim or diaphragm. So, endostome is attached to the rim and exostome is attached to the annulus is wrong. Both endostome and exostome peristomial teeth are attached laterally to the rim or diaphragm, and they closes the openings of spore sacs. So, choose the incorrect statement with reference to Funaria is second option. Endostome is attached to the rim or diaphragm and exostome is attached to the annulus in capsule is wrong. In Funaria, 80 antherozoids are produced in an antheridium. Total number of reduction divisions that occur in that antheridium is, in Funaria, 80 antherozoids are produced in an antheridium. Total number of reduction divisions that occur in that antheridium is haploid. There is no question of reduction divisions. Androgonial cells, androcyte mother cells, androcytes. Metamorphosis of androcytes into antherozoids is the concept in the antheridium. So, because it is haploid, no question of reduction division. So, answer for this is 0. In Funaria, antherozoids are produced in an antheridium. If 80 antherozoids are produced in an antheridium, total number of reduction divisions that occur in an antheridium is, everybody will definitely confuse with this. By 80 by 4, they write 20, but here the answer is 0. Cross fertilization contrivance of Funaria. Funaria is monoecious. Funaria is autoecious. Both female and male reproductive structures are present on the same plant. But still self fertilization does not occur. Only cross fertilization occurs due to protandry. Protandry means maturation of antheridium, maturation of male reproductive structure antheridium much before archegonium is protandry. So it is not protogyny, it is not dioecious, it is not self-sterility, it is protandry. Here andrecium and gynecium will not come. Maturation of male reproductive structures antheridium much before the female reproductive structure archegonium. That's why in Funaria self-fertilization is prevented and only cross-fertilization takes place. Male gametophytes of Funaria are. Male gametophyte means anthrozoids. It comes from antheridium. From androgonial cells to androcyte mother cells, from androcyte mother cells to androcytes, and androcytes become anthrozoids. Sickle-shaped multiflagellate is wrong. Two spirally coiled biflagellate is wrong. Sickle-shaped non-flagellate is wrong. Sickle-shaped biflagellate is correct. So, every anthrozoid in Funaria resembles like this. It is sickle-shaped with two flagella. So, sickle-shaped with two flagella all comes out as a group, fell into the surrounding moisture and by chemotaxis reaches the archegonium and perform fertilization. So, answer is sickle-shaped biflagellate. Funaria is a bryophyte. It is an embryophyte. Tracheophyte is wrong because bryophytes are atracheophytes. Vascular tissues are completely absent. It is a tracheophyte and spermatophyte is completely wrong. Funaria does not produce any seed-like structures. Non-embryophyte itself is wrong. Non-embryophyte, atracheophyte is correct, but non-embryophyte is wrong. Why? Because for the first time in plant kingdom, zygote undergoes mitosis and produce embryo. So, the correct answer is atracheophyte and embryophyte. So, Funaria is atracheophyte where vascular tissues are absent and embryophyte for the first time embryo is formed. Peristomial teeth of Funaria helps in spore dispersal, are arranged in two holes. They are 16 in each hole. So, peristomial teeth in Funaria capsule, they are surrounded by capsule and peristomial teeth number will be 32. They are arranged in two groups of 16 each. Outer 16 are called outer exostome, inner 16 are called inner endostome. Exostome and endostome are triangle in shape, but exostome shows radial and tangential wall thickenings. Radial and tangential wall thickenings. It is hygroscopic in nature. This will not show radial and tangential wall thickenings and it is also not hygroscopic in nature. Peristomial teeth play a vital role in the mechanism of dispersal of spore in Funaria capsule. So, peristomial teeth in Funaria are helping in spore dispersal, are arranged in two holes and each hole shows 16 is correct. So, fourth option 1, 2, 3 are correct. Venter of Archegonia has Funaria Archegonia has double layered jacket correct, egg cell correct, but neck canal cell is wrong. Why? Because neck canal cells are present in the neck, not in the venter. So, double layered jacket, egg cell are correct. So, answer is 1 and 2 are correct. 1 and 2 are correct. Sugary mucilaginous substance of Archegonia is formed from. Sugary mucilaginous substance are formed from. In Funaria, once the Archegonia is formed, the ventral canal cell and neck canal cells will be there. The ventral canal cell disintegrates, the neck canal cell disintegrates and forms a mucilaginous substance with sugars, accumulates at the tip of the neck. So, because they are accumulated at the tip of the neck, they attract the anthrozoids. So, sugary mucilaginous substance of Archegonia is formed from, not from egg cell, because egg cell remains for fertilization. It is from the disintegration of all the neck canal cells and venter canal cell. So fourth option 2 and 3 are correct for this. Now this is a matching one. Match the following and identify the correct match. List 1 trabeculae, haploid columella, 4 cover cells, 2 cover cells. Fineria capsule, Anthridium of Fineria, Sporangiophore, Archegonium of Fineria and Fineria protonema. A1. Fineria capsule contains trabeculae, small stalk-like structures, chlorophyllous in nature, uniseriate cells connecting the outer 3-layered spore sac with assimilatory tissue. In between every 2 trabeculae, air spaces are present. Haploid columella. Haploid columella is found in sporangiophores. Sporangiophore, above the sporangiophore, sporangium will be there and you will find a haploid columella. Then 4 cover cells. 4 cover cells are found in Archegonium of Fineria. At the tip of Archegonium of Fineria, definitely 4 cover cells are present. Then 2 cover cells. 2 cover cells are present in Anthridium of Fineria. So, trabeculae found in Fineria capsule. Haploid columella is found in sporangiophore because sporangiophore ends as sporangium which contains haploid columella. Sporangiophore contains capsule and capsule contains a central special haploid tissue called columella. Then 4 cover cells are found in Archegonium of Fineria and 2 cover cells are found in Anthridium. So, A1, B3, C4, D2. Fourth option. This another matching. Match the following and identify the correct match. Capsule, foot, theca, sporangium. Urn shape, barrel shape, sickle shape, dagger shape, pear shape. The shape of this. So A5 capsule. Capsule is pear shape. It appears like a almost egg shaped structure. So pear shape. B foot. Foot is always dagger shape. Dagger is like a knife and foot is dagger shape. Then theca. Theca is urn shape. Theca is urn shape. And sporangium is barrel shape. So capsule is pear shape, foot is dagger shape, theca is urn shape and sporangium is barrel shape. So answer A5, B4, C1, D2. First option. This is another matching one. List 1, list 2. Protonema, gametophore, anthridia, archegonia. Buds of chloronema, extra-axillary branch, apex of main branch, spore and apogamy. It is also first option because A4 is protonema comes by the germination of spores. Every spore contains outer exospore, inner endospore. Special feature of the spore is contain chloroplast. Once it germinates, it produces either one germ tube or 2 germ tubes which converts into juvenile stage in Fineria called protonema. So protonema develops from the spore. B1. Gametophore always comes from buds of chloronema and a special pigment called or special hormone called bryokenin helps in the formation of gametophore from the buds of chloronema. So buds of chloronema definitely gives rise to gametophores. Then anthridia. Anthridia is present at the apex of main branch. Anthridia is always present at the apex of main branch. Archegonia are present on the extra-axillary branch apex. Why it is extra-axillary? Because in Fineria, branches will not come from the axils, that is in between stem and the leaf. They come from the base of the leaf, so extra-axillary branch. Archegonia are always formed at the tip of archegonial branch. So protonema, spore, gametophore, buds of chloronema, anthridia, apex of main branch, main axis, and archegonia, extra-axillary branch apex. So first option A4, B1, C3, D2. This is a 3-column matching. Structure, ploidy, function. Calyptra is haploid and a transpiring shield is correct. Calyptra is haploid, transpiring shield is correct. Paraphysis haploid. Protecting zygote is wrong. Paraphysis does not protect zygotes. They protect just either anthridia or archegonia. Theca, diploid spore production. So theca, diploid spore production is correct. Central diploid urn-shaped portion in Fineria capsule is theca. It contains sporogenous tissue that produce spore mother cells which ultimately gives rise to spores. Now, peristome diploid photosynthesis. Peristome is diploid correct, but it is not green in color and performing photosynthesis. So, name the two correct combinations means Calyptra haploid transpiring shield is correct. Theca diploid spore production is correct. Paraphysis haploid protecting the zygote is wrong. Peristome diploid photosynthesis is wrong. So, the correct combinations are 1 and 3. Here the option is 4. This is another 3-column tabular form. Structure, part of the stage and function. Pericardial leaves, gametophyte protection to zygote is wrong. Cover cells, sporophyte protection to the neck of archegonium is wrong. Sporophyte, archegonium will not present in sporophyte, it is present in the gametophyte. Tubers, gametophyte, vegetative propagation is correct. What are tubers? Special subterranean resting buds formed on rhizoids, sometimes on colloids, helping in vegetative propagation are called tubers. So, tubers are found in the gametophyte, correct. Helping in vegetative propagation is correct. Seta, sporophyte, spore dispersal. Seta is present in the sporophyte, foot seta and capsule. By twisting and jerking movements of seta only, dispersal of spore takes place. So, the answer is 3 and 4. 1 and 2 are wrong. Pericardial leaves, gametophyte, protection to zygote is wrong. Cover cell, sporophyte is wrong. Cover cell must be gametophyte because they are present in the archegonium. So, answer is tuber, gametophyte, vegetative propagation. Seta, sporophyte, spore, dispersal. 3 and 4 are correct. Another 3-column tabular form. Structure, stage to which it belongs and function. Rim, sporophyte, point of peristome attachment, correct. Rim or diaphragm is present in the diploid sporophyte and it is the point of attachment of peristome. Peristomial teeth are attached laterally to the rim or diaphragm. Annulus, gametophyte, breakage of capsule. So, annulus is present in gametophyte and breakage of capsule. Trabeculae, sporophyte, spore production. Trabeculae, sporophyte, spore production is wrong. Protonema, gametophyte, antheridial production is wrong. So, 1 and 2 are correct. So, 1 and 2, that is second option is correct. Next, zyma. Structure is useful for reproduction, formed on, involved in the type of reproduction. Zyma, injured portions of gametophore, vegetative reproduction. Injured portions of gametophores is wrong. They are present on the roots, they are rhizoids, stem and leaf. Tubers, rhizoids, vegetative propagation. Tubers are formed only on rhizoids and they help in vegetative propagation. Secondary protonema, injured portions of gametophyte, asexual, wrong. Secondary protonema formed from injured portions of gametophyte only, but they are not helping in asexual reproduction, they are helping in vegetative propagation. Archegonia, female branch sexual reproduction. Archegonia is present at the tip of the female branch only and it definitely helps in sexual reproduction. So, for this 3-column matching form, answer is 2 and 4, that is 4. Arrange various gametophytic stages of Funaria in sequence. Arrange various gametophytic stages in Spirangia in. So, first is 3, gametophore. Gametophore is formed. Then protonema formation. Then spore. Finally, sex organ formation. So, protonema formation, gametophore, spores. From the spore, protonema will come and protonema is gametophore and it gives rise to sex organ formation. So, 3, 4, 2, 1. Second option. Arrange the following correct sequence of their formation. Anthrozoite, androgonial cell, androcyte mother cell, androcyte. First androgonial cells are formed. Then androcyte mother cell is formed. Then androcyte is formed. Finally, anthrozoid is formed. So, androgonial cell first, androcyte mother cell next, androcyte then and finally anthrocyte. So, answer is 2, 3, 4, 1. This is the correct sequence of formation of various structures during anthrozoite formation in Funaria. Now, a special assertion and reasoning. Neutral prothali reproduce by apogamy. Neutral prothali reproduce by apogamy. Apogamy is the development of gametophyte from sporophyte without involvement of spore. Apogamy and apospory. There are two extrasexual methods are present in Funaria and Pteris. Apogamy, apospory and apogamy. Apospory means development of haploid gametophyte, development of diploid gametophyte, development of diploid gametophyte from diploid sporophyte, diploid gametophyte from diploid sporophyte without reduction division or meiosis. So, important concept here is diploid gametophyte. Development of diploid gametophyte from diploid sporophyte without reduction division or meiosis is apospory. Apogamy, development of haploid sporophyte. This is special. Sporophyte will not be haploid, but here it is haploid. Haploid sporophyte from haploid gametophyte, haploid sporophyte from haploid gametophyte without the formation and fusion of gametes. These two are special reproductive methods found in Fineria and Pteris. Apospory, apogamy. Apospory is development of diploid gametophyte from diploid sporophyte without reduction, division or meiosis. Normally gametophyte is haploid, but here haploid form. Apogamy, development of haploid sporophyte from haploid gametophyte without the formation and fusion of gamete. Normally sporophyte is diploid, but here haploid sporophyte is formed. So, here the assertion reasoning is neutral prothalae produces or reproduced by apogamy, correct. Neutral prothalae means reproductive structures are not formed. So, definitely apogamy. Apogamy is the development of gametophyte from sporophyte without involvement of spores. Development of gametophytes from sporophyte. No, it is development of sporophyte from gametophyte without the involvement of formation and fusion of gametes. So, A is correct, R is false. Pterosaurus is a mixed type. Pterosaurus is a mixed type is correct. Pterosaurus contain both sporangia and gametangia. Mixed saurus because it shows different stages of sporangia in them. Different stages of sporangia means some sporangia are well developed, some sporangia are still developing, some sporangia are liberated spores and many other structures like this. But Pterosaurus is a mixed type is correct. Pterosaurus containing both sporangia and gametangia is wrong. Pterosaurus contain only sporangia, it does not contain gametangia. So, Pterosaurus is a mixed type, correct. Pterosaurus contains both sporangia and gametangia is wrong. So, answer is A is true, but R is false. The leaf traces and root traces of Pteris are present in the leaf traces and root traces. When you take the TS of rhizome in Pteris, it will be like this: irregular outgrowth or irregular outline with outer thick cuticle. Then inner to it a very prominent sclerenchymatous hypodermis. Then total ground tissue, parenchymatous ground tissue. In the ground tissue you will find vascular bundles like this. The central vascular bundles are larger in size, lesser in number. Peripheral vascular bundles are smaller in size, more in number. This is called dictyostele and each stele is called meristele. This is called dictyostele and each stele is called meristele. If you carefully observe the cortex, it shows special leaf traces and root traces. So, leaf traces is the leaf vascular bundle of the leaf, root trace is the vascular bundle of the root. So, when you clearly observe the TS of rhizome, leaf traces and root traces are not found in hypodermis of rhizome, they are not found in endodermis of rhizome, they are not found in the meristele of rhizome, but found in the cortex of rhizome. Pteris antheridial wall consists of one ring cell and two opercular cells. One ring cell and two opercular cells is correct. Is wrong. Two ring cells are present. First ring cell, second ring cell and two opercular cells. Two rings of cells and one opercular cells. Two rings of cells and one opercular cells is correct. Because Pteris antheridium resembles like this. It shows one opercular cell, first ring cell, second ring cell. Like this. Another cell will be there. This is stalk cell, but it is not belonging to the antheridium. It belongs to female what you call prothallus, tissue of prothallus. So here the answer is for 2 only, 2 ring cells and 1 opercular cells, 3 lid cells wrong, 1 ring cell and 2 opercular cells is wrong. Now, in Pteris both prothallus and sporophytes possess roots. In Pteris both prothallus and sporophyte possess roots. In Pteris both sporophyte and gametophyte are independent. In Pteris both prothallus and sporophyte possess roots is wrong. Prothallus produces rhizoids. Prothallus does not produce roots, it produces rhizoids. Sporophyte produces adventitious roots. So, in Pteris both sporophyte and gametophyte are independent. In the total plant kingdom, the only plant group where the independent gametophyte and sporophyte are present is Pteris. So, A is false, R is true. Fourth option. The common character shared by both Pteris and Funaria. Common character shared by both Pteris and or Funaria and Pteris. Rhizoidal gametophytes both will show rhizoidal gametophytes. Vascularized sporophytes are found only in Pteris, not in Funaria. Heteromorphic alternation of generation. Sporophyte and gametophyte are entirely different but perfectly alternate with each other. So, heteromorphic alternation of generations is correct. Haplodiploidic life cycle. In Pteris it is diplohaplontic life cycle, in Funaria it is haplodiploidic life cycle. So, vascularized sporophytes are found only in pteridophytes. Haplodiploidic life cycle is found only in Funaria. So, the common characters shared by both Funaria and Pteris are rhizoidal gametophytes, heteromorphic alternation of generations, that is A and C. Third option. Arrange the following structures in Pteris in correct increasing order based on their number. Maximum number of spores produced by sporangium is 48. Sori in each pinnule is 2. Reduction divisions that occur in each sporangium is 12. And spores produced by vegetative leaflet of frond is many. So, answer is 4. Spores produced by vegetative leaflet of frond is 0. Then 2. Sori in each pinnule is many are 2. 3. Reduction divisions occur in each sporangium is 1. 1 reduction division results in the formation of 48. And finally, maximum number is 48. So, 12, 48, 2 and 0. So, answer is 1, 4, 2, 3, 1. It is an assertion reasoning. The spore liberation in Pteris occurs by sting mechanism or catapult mechanism. It is correct. The sporangium of the Pteris contains special ring-like cells called annulus. Annulus, when loses water, it contracts. When it contracts, it bends. When it bends, it makes the lip cells of stomium to move wide apart from each other with a gap in between themselves. At one point of time, annulus suddenly absorbs moisture. Suddenly it comes to normal position, does not give ample time for the lip cells to close. So, all these spores get liberated. This bending down of body and coming to the normal position by the activity of annulus is popularly called catapult mechanism or sting mechanism. So, spore liberation in Pteris is occurring by catapult mechanism or sting mechanism is very much correct. The spores in Pteris are carried away by wind. Spores in Pteris are carried away by wind is also correct, but not because of catapult mechanism or sting mechanism. Spores are carried away by wind because they are smaller in size and easily carried, they are carried away by wind. So, both are correct, but R is not the correct explanation. Answer will be both A and R are correct. R does not explain A, R is not the correct explanation of A. Calculate the total number of male gametes and number of new sporophyte produced from the prothallus of Pteris with 20 antheridia and 10 archegonia respectively. 20 antheridia means each antheridium produces 34 antherozoids. So, 640 antherozoids. But prothallus, how many antheridia contains? Nobody bothers. How many archegonia contains? Nobody bothers. But only one sporophyte comes from each prothallus. So, calculate the total number of male gametes and the number of new sporophytes formed from a prothallus of Pteris with 20 antheridia and 10 archegonia respectively means even though 10 archegonia are there, 20 archegonia are there, 40 archegonia are there, only one archegonium is functional, all the others are abortive. So, only one sporophyte is formed. So, answer for this is 640 and 1. 640 antherozoids are formed and one sporophyte is produced. Venation in Pteris is described as, when you observe a frond in Pteris, When you observe a frond in Pteris, it shows a prominent midrib. From the midrib, lateral veins are produced. And all lateral veins speciality is they are forked at the tip. They are forked at the tip. This type of venation is called open dichotomous venation. So, lateral veins always forked at the tip. This type of venation is called open dichotomous venation. Pinnate reticulate is wrong. Palmate reticulate is wrong. Pinnate reticulate is wrong. Palmate reticulate is wrong. Pinnate parallel is wrong. The answer is open dichotomous venation. Venation in Pteris is described as open dichotomous venation. Arrange the following cells in Antheridium in ascending order. Ascending order means from base to apex. First ring cell, second ring cell, basal cell, cap cell. Basal cell or stalk cell is formed from tissue of prothallus, that will be first. Then first ring cell and first 3 will come, then 1 will come, then second ring cell, then cap cell. So, correct order is at the base, basal cell or stalk cell formed from prothallus, tissue of prothallus, then first ring cell forms the base of the antheridium, then second ring cell forms the sides of the antheridium, and cap cell forms the topmost portion of antheridium. So, answer is basal cell, first ring cell, second ring cell, cap cell. So, second option 3, 1, 2, 4. 3, 1, 2, 4. Now, arrange the following events in a sequence in the life cycle of Pteris. Meiosis, sporangium formation, prothallus, oogamy. First sporangium formation, then meiosis. Sporangium formation takes place in fronds. Then in sporangium meiosis takes place, 48 spores are formed. Then prothallus, then oogamy. Prothallus, then oogamy. Then prothallus, then oogamy takes place. That is, answer is 2, 1, 3, 4. Because first sporangium formation, then meiosis, then spores germinates to produce prothallus, then prothallus produces male and female reproductive structures, and finally oogamy takes place in sexual reproduction. So, 2, 1, 3, 4 is correct sequence in the life cycle of Pteris. Arrange the following in a sequence of the last cell of sporophyte and first cell of gametophyte, last cell of gametophyte and first cell of sporophyte. So, first cell of gametophyte is first cell of gametophyte, poor mother cell. Then last cell of gametophyte, spore. Then last cell of gametophyte is gamete, and first cell of sporophyte is zygote. So, answer is 4, 1, 3, 2. Second option is the answer. This is a 3-column tabular form belonging to Cycas. Leaflet, dichotomous venation, spindle shape. Wrong. Root, sclerenchymatous inner cortex, diarch condition. Sclerenchymatous inner cortex, diarch condition is correct. Stem, dictyostele and corm. Dictyostele and corm is wrong. It is not corm, it is rhizome. Sporophyll falls into paraphysis. It is 2 correct items, paraphysis and falls into both appear in the sporophyll. So, answer is 2 and 4, second option, fourth option. This is another 3 column structure, feature 1, feature 2. Spore, triangular, trilete. Trilete means a triradiate special stalk is present. Archegonium, flask shaped single neck canal cell, correct. Antheridium, double layered jacket, 12 androcytes is wrong. 12 and spore mother cells will be present. Prothallus, dorsiventral, stellate chloroplast is wrong. Chloroplast is discoid, not stellate. So, answer 1 and 2. Spore, triangular in shape, trilete. Trilete means a triradiate arm is present. Archegonium is flask shaped but single neck canal cell. Archegonium is flask shaped, single neck canal cell. That is also correct. So, 1 and 2 are correct. Antheridium, double layered, 12 androcytes is wrong. In total 32 androcytes are present. Prothallus, dorsiventral, stellate chloroplast is wrong. It is discoid chloroplast. So, here correct combinations are 1 and 2. Wrong combinations are 3 and 4. This is another 3-column tabular form. Structure, position, jacket. Archegonium, ventral position, single layered. It is not single layered. Antheridium, ventral position, single layered. It is correct. Sporangium, dorsal side, double layered. Correct. Adventitious roots, ventral side present. Wrong. So adventitious roots are not present. They are present rhizoids. So second and third options are correct regarding this. Structure, position, nature of jacket. Second option is correct. 2 and 3. Organ of Pteris, character 1, character 2. Rhizome, dictyostyle, endarch is wrong. It must be mesarch. Root, sclerenchymatous inner cortex, diarch and exarch, correct. Gametophyte, spherical in shape, discoid chloroplast, wrong. It is not spherical, it is heart shaped. Leaflet, hypostomasus, amphicribal vascular bundles. Amphicribal means xylem at the center surrounded by phloem on all sides. So answer is 2 and 4 are correct options. 1 and 3 are wrong options. Then part, character 1, character 2. Tears of Pteris root. Tears of Pteris roots, diarch steel, presence of medulla wrong. Tears of Pteris rhizome, dictyostyle, endarch xylem is wrong. Again it is mesarch. Tears of Pteris rachis, bicelled ramenta, mesarch xylem correct. Ramenta are always two-celled. Tears of Pteris leaflet, hypostomatous condition, amphicribal vascular bundle correct. So answer is 3 and 4. Now match the following, identify the correct match regarding Pteris. Receptacle, seta, ring cells, capsule. Jacket of Antheridium, cover cells, spore sac, pedicel and placenta. A5. Receptacle or placenta are present in the Cynosaurus or Saurus. Seta. B4. Pedicel. The other name of seta is stalk and also pedicel. Ring cells. 1. Jacket of Antheridium always contain first ring cell, second ring cell and 2 capsules. Then capsule. Capsule contains spore sac. In the capsule, 2 to 3 layers of spore sac, externally surrounded by single layered spore sac, internally surrounded by one layered spore sac. So answer is A5, B4, C1, D3. First option. Match the following and identify the correct match. Elliptical, chordate, U-shaped and oval. Vascular bundles of rachis, shape of rhizome in Pteris, rachis in Pteris, vascular bundle of rhizome and prothallus. Answer is 3. A4. Elliptical. Vascular bundle of rhizome is elliptical. Then B5. Chordate. Prothallus is always chordate shaped with broad anterior region showing U or V shaped notch. So prothallus of Pteris always shows a structure like this. It shows anterior portion and it shows posterior portion. Anterior portion shows U or V shaped notch. Posterior portion is pointed. From the posterior portion, many rhizoids are produced. It shows central cushion-like portion where many tissues aggregate to form a cushion-like. On both sides, it shows single-celled thick wings. Meristematic tissues are present at U or V-shaped notch. During reproduction, archegonia develop from the cushion-like portion near U or V-shaped notch. Antheridia are produced in between the rhizoids towards the posterior end. Both antheridia and archegonia are formed on the ventral side, and as its prothallus comes from a single spore or homosporous spore, it is monoecious. It shows both male and female reproductive structures in the same plant. Then U-shaped. U-shaped is vascular bundle of rachis is U-shaped. C1. Then oval. D3. D3 is rachis. Rachis in Pteris is oval in shape. So third option. Match the following and identify the correct match. Androcytes per antheridium will be 32. Jacket cells per archegonium will be 16. Jacket cells, 4 rows of jacket cells arranged in 4-celled height. So definitely they are 16. Spores per spore sac is 8. Spores per spore sacs comes is 8. Not 8, 48. Spores from per spore sac is 48. Every sporangium produces 48. Cells in developed embryo is 8. So answer is 1. A3. Androcytes per antheridium is 32. B4. Jacquet's pulse per archegonium is 16. 4 rows of neck cells in 4 celled height means all the 4 sides 4 celled height, 4 cells means 4 into 4 16. Spores per spore sac is 48. C5. Every sporangium in Pteris produce 48 spores, and that means spore mother cells present in the sporangium is 12. Cells in developed embryo is 8. First, how the developed embryo will start? It will start with a structure like this. First division is transverse division. Next division is transverse division but perpendicular to the first division. How many cells are formed? 4 cells are formed. Now, in each half of this cell, another transverse division takes place. How many cells are formed? 8 cells are formed. The most important aspect in Pteris is whenever the organogenesis or morphogenesis takes place, it takes place only at 8 celled stage. So, coming to the number, androcytes per antheridium will be 32. Jacquet cells per archegonium will be 16. Spores per spore sac will be 48. And cells in a developed embryo will be 8, because in 8 celled stage only the embryo shows morphogenesis or organogenesis. So, answer 1. Arrange the following events in a proper sequence that occur in the life cycle of Pteris. Sporogenesis, development of antheridia, maturation of archegonia, arrest of prothallus growth. First sporogenesis, spore formation, then development of antheridia, that is when prothallus is still very young, antheridia are formed, then maturation of archegonia which occurs later, and arrest of prothallus growth, development of new plant. So, for this, arrange the following events in a proper sequence that occur in the life cycle of Pteris is 1, 2, 3, 4. Assertion reasoning. The stele in the stem of Pteris is dictyostele. The stele is split into meristeles in the stem of Pteris. So, in the rhizome of Pteris, definitely the stele is popularly known as dictyostele. Why it is dictyostele? Because the total stele is cut into small bits called meristeles. So, both A and R are correct. R is the correct explanation of A, or R explains A, first option. Now, Pteris is homosporous. Definitely Pteris is homosporous because all the spores are of single type. That is why each spore germinates and gives rise to monoecious prothallus. That means prothallus showing male antheridium and female archegonium in the same prothallus. Two types of spores are sometimes formed in Pteris. Pteris absolutely does not produce two types of spores. So, A is correct, R is false, first option. Third option, another assertion reasoning. Life cycle of Pteris shows heteromorphic alternation of generations. Life cycle of Pteris shows heteromorphic alternation of generations. Both sporophytic stage and gametophytic stage are present in the life cycle of pteridophyte. Life cycle of Pteris is popularly called heteromorphic alternation of generation is correct. Both sporophytic stage and gametophytic stage are present in the life cycle of pteridophyte is correct, but is also correct but not the correct explanation of A. Why? Because heteromorphic alternation of generations means sporophyte and gametophyte are entirely different structurally, functionally, physically, physiologically, in all aspects, but they perfectly alternate with each other. That is called heteromorphic alternation of generation. So, here A is correct, R is correct, but R is not the correct explanation of A. So, definitely the answer will be both A and R are true. R does not explains A. Second option. Now, this is a matching regarding Cycas. Number of cells just released from pollen grain 3. Number of male gametes formed in the pollen tube 2. Maximum number of archegonia present in the ovule 8, 2 to 8 archegonia. Number of microspores in a tetrad. Number of microspores in a tetrad is always 4. Is always 4. Just a minute. Tetrad. Microspores in a tetrad is 4. Tetrad means 4. So, answer A2. A2 is number of cells in just released pollen grain is 3. B4. Number of male gametes formed in a pollen tube is 2. C5. Maximum number of archegonia in an ovule of Cycas is 2. And number of microspores in a tetrad is 4. D3. So, A2, B4, C5, D3. First option. Now, another matching regarding Cycas. Match the following with reference to Cycas. Algal zone of coraloid root is middle cortex. Always middle cortex is called algal zone. Xylem of coralloid root is always triarch and exarch. Xylem of coralloid root is always triarch and exarch. Hypodermis of leaflet. Hypodermis of leaflet, that is C. Hypodermis of leaflet is always heat screen. Leaflet hypodermis is sclerenchymatous. It functions like a heat screen. Then xylem of leaflet is pseudomesarch in condition. Why pseudomesarch? The protoxylem of centripetal xylem is surrounded by metaxylem of centrifugal xylem. So, two types of xylem are present, diploxylic condition. So, centripetal xylem's protoxylem is surrounded by centrifugal xylem's metaxylem. So, it is mesarch, pseudomesarch. It is not real mesarch, it is pseudomesarch. So, algal zone of coralloid root is triarch and exarch condition. Xylem of coralloid root, algal root of coralloid root is middle cortex. Xylem of coralloid root is triarch and exarch. Hypodermis of leaflet is heat screen. It definitely functions like a heat screen and provide protection to the leaflet from hot sunlight. Xylem of leaflet always shows pseudomesarch condition. So, second option A2, B4, C5, D3.